This video is about B4, the processes of life. We'll be looking at photosynthesis, so how plants make their own food, transport in plants, which looks at diffusion and active transport, inside cells, which looks at animal, plant, yeast and bacterial cells, respiration, enzymes and factors that affect them, and lastly we'll be looking at how we measure plant distribution in certain areas. So, first we'll look at photosynthesis. So if I start to draw a plant here, growing from the soil, and there are the roots, I can show you an enlarged version of a leaf, and the leaf is essentially the gas exchange organ of the plant, in the same way the lungs exchange gas in animals. So we see that carbon dioxide is taken into the leaf at certain parts and oxygen is released from the leaf. We can also see the roots are taking in water up the plant, and sunlight is being absorbed by the green parts of the plant, which contain chlorophyll, and that's mainly the leaf. So we can show that in an equation, or a word equation. So carbon dioxide plus water goes to glucose plus oxygen, and it needs light for that reaction to happen, and that's photosynthesis. So glucose is the useful product that the plant wants to make, that's the food. We don't call it food though, we call it glucose or sugar. And the oxygen is the waste product for the plant. It's still useful for us, but we call it a waste product for the plant. So why do plants need to use glucose? Well, first of all, they need glucose for storage of energy, and that's either in starch form, which is a carbohydrate, or oils, which are a type of fat. Plants need it for respiration, so plants have to respire too to get energy, and the energy is obtained by respiration. Proteins are made in plants to grow and repair, and that requires glucose. And also cellulose, which is the cell wall of a plant, is very strong, and it gives the plant cell structure, and that requires glucose to make cellulose. So now let's look at how different variables affect the rate of photosynthesis. So first of all, we should notice that the equation there contains the variables that affect photosynthesis, carbon dioxide and water, for example. So more carbon dioxide means the rate of photosynthesis should really increase, and of course less carbon dioxide means less photosynthesis. The same can be applied to water. The more water there is, we should assume there'll be an increase in photosynthesis, and of course if there's more light, there should also be an increase in photosynthesis. But let's look at this in more detail now, and look at which factors are more important. So, if I plot a graph of the percentage of carbon dioxide against the rate of photosynthesis, and if we plot two lines on the graph, one for low light levels and one for high light levels, we see the following pattern. So this line here is when there's a huge amount of light, and we see that as we increase carbon dioxide, the rate of photosynthesis increases to a certain point and then levels off. So I've described the trend there, and to explain it, I could say of course that more carbon dioxide means there's more reactants for products to form. Now when there's low light, actually the rate of photosynthesis still increases to a certain point, but it's not as high a point. So we call the light the limiting factor. And this means that even if there is a high amount of carbon dioxide at low light levels, there's only a very small increase in the rate of photosynthesis, whereas when you increase the light and the carbon dioxide, there is increased rate. So now let's see how temperature affects rate of photosynthesis at different light levels. So again we'll do high and low. That's the low light level line, and this one is the high light level line. And we see that actually at the low level of light, if you increase the temperature, it generally has no effect on the rate of photosynthesis until a certain point where it decreases the rate, and that's because the enzymes are denatured. Whereas in the high light level line, increasing temperature first of all increases the rate of photosynthesis to a certain high point, and then of course after a certain temperature it then massively decreases the rate of photosynthesis. So we see an increasing temperature does increase photosynthesis rate until the enzymes denature. So now let's look at transport in plants, and we'll look at diffusion first of all. So if I draw some particles in a very small space, so a very concentrated amount of particles, I can show green arrows to say that the particles are moving in random motion, bumping into each other, and after a certain amount of time goes by, they will eventually spread out due to that random motion. So they'll go from a higher concentration to a low concentration simply randomly, because there's more chance of that happening than the reverse of them going into a very small space. And we see that if we increase temperature, we increase the movement of these particles and the speed of their movement, and therefore increase the rate of diffusion. Now, let's look at osmosis. Osmosis is basically a very special type of diffusion involving water. And if I draw a semi-permeable membrane, which lets some things through and not others, for example, it might let small particles of water through, but not large particles, we see the following. So those blue dots are water, and they can move back and forward. And if the concentration of water is the same on the left and the right hand side, the net flow is 0. But if I add some molecules of sugar on the right hand side there, which can't fit through the membrane, well, now we've got a less concentrated amount of water on the right hand side and a higher concentration of water on the left. So the water will go to the right, from a high concentration of water to a low concentration of water. But if I add more particles on the left hand side now, of course, there's now less water concentration on the left hand side, and water will then reflow from the right hand side, which is high in concentration of water, to the left hand side. So osmosis simply means water diffusing randomly from a high concentration of water to a low concentration of water through a semi-permeable membrane. Now both processes involve random motion, and both are passive because they require no energy to happen. So let's now look at another process. And if I draw a plant growing in some soil, and the soil contains minerals which are usually metal ions, such as magnesium 2+, which the plant needs to grow healthily. The plant needs to uptake these minerals, but the minerals are usually quite large compared to water. They're usually charged, so they're ions, and therefore they're hard to take up. So passive diffusion wouldn't really work very well here. Now also, sometimes these minerals are taken up against the concentration gradient. So there's often more minerals inside the plant than the outside of the plant. So if the plant needs to get more minerals, it can't do it by diffusion, because diffusion means going from a higher concentration to a low concentration, when actually you're trying to go from a low concentration to a high concentration. So what you need to do is use some sort of protein to pull the minerals through the cell membrane into the cell, and that requires energy. So this energy being used allows that movement against the concentration gradient, and that's called active transport, and it's not random and it's not passive. So it's different to osmosis and it's different to diffusion. Now you do need to know a bit about minerals themselves. So potassium, for example, is used to make proteins for growth. Nitrogen is not so much a mineral, but it is something used in plants, and it's used for rapid growth and photosynthesis. Phosphorus is used for photosynthesis as well, and magnesium is used to create the chlorophyll, which is the pigment inside chloroplasts that absorbs light for photosynthesis. So minerals are very important in plants, and without them they can't grow. Now a hyperaccumulator is a plant that takes up lots and lots of minerals from the soil, including harmful minerals such as lead, Pb. And as it does this, it locks those minerals inside its cells, and it removes harmful minerals from soil. So it effectively reclaims the soil that was previously dangerous to grow plants on. And these are what hyperaccumulators are used for. So now let's look at inside cells. So first of all, we'll draw an animal cell, and I'm drawing a cell membrane. Remember, animal cells do not have a cell wall. There's a nucleus and a surrounding nuclear envelope, which contains nuclear pores to allow information to be exported. Around the nuclear envelope and the nucleus is something called rough endoplasmic reticulum, or ER, which contains these tiny structures on top of it to make it rough, called ribosomes. Smooth ER doesn't contain the ribosomes, and the ribosomes are used to make proteins. Now those brown diagrams there are mitochondria, which are used to release energy from the cell. We also have cytoplasm, which is the liquid that things can be dissolved in to react. Now a plant cell has some features that are similar and some different features to an animal cell. First of all, it has a cell wall made of cellulose, which gives the plant its structure, a cell membrane to allow selectively allow some chemicals into and out of the cell, mitochondria again to release energy. Now those blue things there are chloroplasts, and they contain chlorophyll, which traps light to be used for photosynthesis. You've also got a large vacuole there, which stores sap, a nucleus and a nuclear pore as is also in the animal cell. And I haven't shown them here, but it also contains the rough and the smooth ER. So, now let's look at a bacterial cell, which are around 10 times smaller than animal and plant cells. And this bacterial cell contains a flagellum, which is used for movement, but of course some bacteria might have 2, 3, 4, 5, or sometimes no flagellum. It also has a single chromosome, which is made from DNA, and it's not contained in a nucleus because bacterial cells do not have nuclei. It contains cytoplasm and 3 ribosomes, but it doesn't have any rough or smooth ER. It just has 3 ribosomes, and it usually has a cell wall made of peptidoglycan and a cell membrane. Now, yeast are actually larger, but not as large as an animal cell, but they're larger than a bacterial cell, and they contain both a cell wall not made of cellulose and also a cell membrane again. It contains 3 ribosomes like bacteria does, but it also has a nucleus which bacteria do not have. It has mitochondria, which also bacteria do not have, and the mitochondria release the energy. And of course it has cytoplasm and large vacuoles. So you should be able to compare those 4 types of cells, but also explain what each part does. And please never say that the nucleus is a brain of a cell. It's not the brain of a cell. Don't say that. Now, let's look at respiration. So we've got aerobic respiration, which means with oxygen. And the word equation is glucose, which is the food, plus oxygen, goes to carbon dioxide plus water, which we exhale. And I'm drawing you there a very simplified version of glucose, which is C6H12O6. And if it's added to 6 oxygen molecules, it produces 6 carbon dioxide molecules plus 6 H2O molecules. You do have to know that for the higher tier. So that's the respiration equation. And I'm drawing there an example of if you set fire to glucose, you'd also get carbon dioxide and water given off. And that's called combustion. So combustion has the same equation as respiration, but the difference is combustion releases lots of energy in a small time, whereas respiration releases energy slowly over a larger time scale. And we know that's obvious because if we did have combustion going on instead of respiration, we'd be on fire. So anaerobic respiration is actually without oxygen, and there are different types of anaerobic respiration. So the one that happens in humans is that glucose is converted without oxygen to lactic acid, and it releases about 2 kilojoules per gram of glucose, whereas aerobic respiration releases about 16 kilojoules per gram of glucose. So aerobic respiration is much more efficient, and also anaerobic respiration produces lactic acid, and that's poisonous for our body. So when you do a sprint, the burning sensation is actually lactic acid. So lactic acid needs oxygen to react with it to form carbon dioxide and water, and that's why after you sprint, you're often out of breath because your body's trying to take in oxygen to relieve the lactic acid. Now, yeast have a different reaction. Their glucose is used to make both ethanol, which is alcohol, and carbon dioxide. So carbon dioxide is used to make bread rise, and that's how you make bread, whereas the ethanol is used to make alcoholic drinks. So yeast is a very important industrial microorganism, as its respiration produces 2 useful products, different to human respiration. Now, you do need to know about fermentation. So fermentation is anaerobic respiration, and of course we've just said that yeast produces ethanol and carbon dioxide used in bread and alcohol. Lactobacillus, you have to know, uses sugar and it forms lactic acid like our own human bodies do, and that's enough to make milk turn into yogurt. Another bacteria is called Acetobacter, and Acetobacter can convert wine into vinegar. Now vinegar is actually called ethanoic acid. The old-fashioned name used to be called acetic acid. So Acetobacter converts wine into vinegar for cooking, and those are the types of microorganisms and their respiration you have to know. So let's look at enzymes now. This is an enzyme I'm drawing, and next to it I've shown you how a substrate combines to the enzyme. So the reactant is called the substrate, and the enzyme basically doesn't react with the reactant or the substrate, but it speeds up the chemical reaction without being used up. And the active site is a part of the enzyme that the substrate binds to. So if I now draw that reaction happening, the substrate is now broken apart to form 2 pieces. That's one example of how enzymes can work. That's called a catabolic or breaking down reaction. And this model is the lock and key model, and the idea is that the enzyme is the lock and the substrate is the key, and different substrates have different shapes and fit different enzymes. So that substrate there has a different shape, it can't fit into the enzyme, and therefore it can't react with the enzyme, essentially. And that's the lock and key model. Now if I zoom into the enzyme, what I can see is a long chain of amino acids making a protein, because enzymes are proteins. And those different amino acids kind of combine together to form a really complex 3D structure that I'm drawing there, if I zoom out slightly. And that has an active site, or a certain shape, that fits around a specific substrate. And that complex 3D structure, made of amino acids, gives the enzyme its capability. So different enzymes have different complex 3D structures. Now that structure is affected by both temperature and pH, and we'll see how that happens. So if I plot a graph showing temperature against the enzyme activity, you get this shape. So as you increase the temperature from 0 to 40 on this particular enzyme, the rate increases, but after a certain temperature, which is about 45 here, it then suddenly decreases because the enzyme becomes denatured. So after a certain high temperature, the enzyme no longer works, it's permanently denatured, and if you increase the temperature before that point, the rate increases. So I'm drawing a diagram to show why. So the enzyme here is bumping into the substrates, and the faster the substrates move, the more collisions happen. So at point A, as you increase the temperature, you increase the kinetic energy of the particles, and therefore you increase the number of collisions. It's just simple chemistry. Now, point B is the optimum temperature. That's where the enzyme is working the most efficiently and the fastest, and therefore it can't go any more efficient than that point, the optimum point. Once you pass that point, what happens is the high energy starts to actually break the structure of the enzyme permanently, and I'm showing you how this happens. So the structure of the enzyme, the 3D structure of the enzyme, changes permanently, and that means the substrates no longer fit the enzyme active site, and therefore it's said to be denatured, and therefore it no longer works effectively or at all. Now let's look at pH against enzyme activity now. So if I do pH from 1 to 13, we get a similar shape and a similar optimum pH. And of course at either side of the pH scale, you will find enzymes work less effectively. And at certain extremes, if you change the pH drastically, you can denature the enzyme, and therefore it will no longer work. But I'm showing you one example of an enzyme there. Actually, different enzymes have different optimum temperatures and different optimum pHs. For example, some work in the stomach very well, some work in the mouth, some work in the intestines. So don't assume that all enzymes have a pH optimum of 7. They don't. So now we'll look at how we can measure plant distribution. And first of all, you can use an identification key. And this basically requires you to ask lots of questions, and depending on your answer, you can find out what species of animal you have. So for example, you might say, does it have fruits, this example of a plant? If it's yes, it might be plant A. If it's not, you might ask a new question. And as you start asking questions, a bit like playing 20 questions, you can narrow down the species that you're looking for. So people that research in the field use an identification key to ask questions to find out what they're actually looking at, so which plant they're observing. So once you know what plants you've got in the field by using an identification key, you want to try and count how many of each you have, or what we call measuring the abundance of a plant. And for this you use a 1 meter by 1 meter quadrat, which is basically a wooden square, and you literally just throw that randomly in the field, and depending on where it lands, you count the number of each species of plant that you can find in that square. And you might throw it a number of times, and each time you throw it, you can measure the coordinates that you've thrown it at, and then you can use certain methods to calculate roughly how much or how many plants you have for the whole field. It's an estimation. Another way of doing it is to do a transect. So similarly, you put quadrats in a long line across the field, and you systematically count the number of each species of plant in each quadrat across the whole line of the field. If it's a very large field, you can actually do it in intervals of, say, 10 meters each. And this method is probably better than throwing the quadrat randomly, because you can measure how gradual changes happen across a field in different conditions. For example, certain parts might be lighter or darker.